Start.
Good morning, children. Here I'm back again with another class of Botany second year, Unit 1, Plant Physiology, and Chapter number 4, Photosynthesis in Higher Plants. Before I begin, I would like to go for this appreciation section, and here I'm going to appreciate these children who submitted their work. S. Akila, R. Krishnaveni, Karishma, Nagasri from Vaira, TSRJC, and Lohita, Abhinasri, Harika from Vangara, TSRJC. Very good children, keep it up, keep the work. And I hope other children are also doing their work in time and sharing their work with their concerned lecturer. And they are also forwarding. And some of the names I'll be carrying for the next session. So today, let's begin with our class. We have completed historical background of the scientist who discovered about photosynthesis process and made our understanding about this life process very easier. Photosynthesis in higher plants. Under this, we have the part 2, that is the site of photosynthesis. We are going to begin with the topic, site of photosynthesis. It is nothing but the place where the photosynthesis actually takes place, that is the chloroplast. In the chloroplast, what is happening? What all the things, what are the internal organization and how it works will be dealt. That means the light reaction and the dark reaction and the reaction center where it is going to trap the solar energy will be dealt. So let us begin with this. Raw materials of photosynthesis. As you all know, water, very important raw material, and carbon dioxide, CO2 from the atmosphere it is taken, and the sunlight and pigments. These four are the raw materials, and these pigments are going to harvest the sunlight energy, and sunlight is having the radiant energy. This radiant energy will be converted to chemical bound energy by utilizing carbon dioxide and H2O, that is water. So in this picture, as you all can see here, we are having a clear cut vision of how the sunlight energy is getting trapped by the green leaves. Meanwhile, the carbon dioxide from the atmosphere is also taken here, and the water from the soil is taken by the xylem vessels and given to the leaf. The green color part of the plant body where the pigments are present. And also they are going to synthesize the glucose, and excess of water will be lost in the form of transpiration, loss of water, as well as transpiration is also very important process. That means in tall trees where the root pressure is not sufficient to send the water to a large height, it is the mode, transpiration is the mode of supplying water to the upper parts of the plant body. So here, this transpiration can also be studied properly in the first chapter. Have a reading about the first chapter for a clear cut understanding. Moreover, here we are saying about water. Water is very, very important factor. This water is going to dissolve some of the essential nutrients present in the soil and supply it. So essential nutrients which are important for the growth of the plant body. About this, the importance is given in mineral nutrition topic, chapter number 2, which is deleted. So have a reading about that lesson 2 and proceed with this chapter. So here, photosynthesis equation is also given. Carbon dioxide 6 molecules is going to react with 12 molecules of water, and in the presence of sunlight, that means by trapping the sunlight energy only, the chloroplast is able to synthesize the glucose, glucose C6H12O6, and it releases 6 oxygen molecules. And one more thing is missing here, that is 6 molecules of water. So this is the photochemical equation that you have learned so far. Now the most important thing, pigments. What is a pigment? Pigment is a molecule which absorbs light of specific wavelength. We know chloroplast. Chloroplast is a structure. Chloroplast is a structure present in plant cell. It is green in color due to the presence of the thylakoid membranes containing the chlorophyll pigment. Here thylakoid membrane will be there, and it contains the chlorophyll pigment. So this particular chlorophyll pigment is very, very, very important. to trap the solar energy or harvest the sunlight energy. And carotenoids and phycobilins, these are also going to harvest. These are accessory pigments. They will also trap the sunlight energy and they will be supplying it to the chlorophyll reaction center. Chlorophyll A is the boss. So here, remember, pigment is nothing but a molecule that absorbs light of specific wavelength. And here, this chlorophyll pigment, it is of several types, 7 types. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E, and bacteriochlorophyll, bacterioviridin. These are the 7 types of this chlorophyll A is very important. And here, chlorophyll pigment will be green in color. That is going to give green color to the plant body. And carotenoids. Carotenoids are 2 types, carotene and xanthophyll. Carotene is red in color, xanthophyll is yellowish in color, yellowish brown in color. And xanthophyll is of 2 types, fucoxanthin and lutein. These are the examples of xanthophyll. And phycobilins. Phycobilins, it is 2 types, phycocyanin and phycoerythrin. Phycocyanin is red in color and phycocyanin is purple in color and phycoerythrin is red in color. So here, this is all about the pigments, but in detail, we have to study the structure of the chlorophyll pigment, what actually it looks like. So in this next slide, we are going to see about that chlorophyll, structure of chlorophyll. Chlorophyll A contains a magnesium ion in the center. That means it is encased in a large ring structure known as chlorine, or it is a head region. And the chlorine ring is a heterocyclic compound derived from pyrrole. And 4 nitrogen atoms from the chlorine surround and bind the magnesium atom. And the magnesium center uniquely defines the structure as a chlorophyll molecule. So here, just I can draw the structure of this chlorophyll for making it easier to understand. Uh, the magnesium will be in the center. Actually, the part of the chlorophyll is having head region and the tail region. So the head region, for example, this is the magnesium in the center, and it will be having 1, 2, 3, 4 here, nitrogen. Okay, so like this, it will be having the nitrogen. So just to make easier, I'll be drawing here. You can see here, first carbon, second carbon will be here for this, and for this nitrogen also. So this is the magnesium. Nitrogen, and here we draw the structure, it will be like this. And for this is the first pyrrole ring of nitrogen, and here we will be having second pyrrole ring, and here the third pyrrole ring, and the fourth one will be here, and one more ring will be there here. Fifth ring is also present here. So 1, 2, 3, number 3, and this is number 4. Remember the number of pyrrole rings, and this is the fifth pyrrole ring. And here the carbon atoms will be placed. Carbon atoms will be placed in the first carbon here, second carbon here, and this is the third carbon, fourth carbon, and fifth and sixth, seventh and eighth, and ninth and 10th. 10 carbons will be there here. And the most important thing why I have drawn here means, in this head region of this chlorophyll pigment structure, you'll see magnesium is in the center. And you know about the pyrrole rings, that is the nitrogen. And in this nitrogen, you can see the position of the carbons, 1, 2, 3. This third carbon, remember this third carbon, for chlorophyll A, at the third position, it is called R position. The R position, third carbon position, methyl group will be attached to the chlorophyll A. whereas aldehyde group will be attached to the chlorophyll B. That is the major difference between chlorophyll A and chlorophyll B structure. So to have a clear understanding about chlorophyll A, why the types are different chlorophyll B. So a simple understanding is given here in this structure of chlorophyll. The magnesium at the center and the pyrrole rings. Okay, you can see the nitrogen here, and here in this pyrrole ring also, this is the first one, second, third, fourth, five pyrrole rings you can see, and the carbon position in this pyrrole ring, first carbon, second carbon, third carbon. This position is very important. That is the R position, or it has the methyl group for chlorophyll A and aldehyde group for chlorophyll B. And the most important seventh is also very important here. Seventh carbon. It is at this seventh carbon. 20 carbons are going to be a chain-like structure that is called the phytol side chain or the phytol tail also in short. So I think you understood the structure of chlorophyll pigment. It has a head, it has a tail. The head will be having the magnesium in the center, and it will be having the pyrrole chain or porphyrin head, or it is also called as tetrapyloric head. Major 1, 2, 3, 4. These are main, and one more is also there, pyrrole here. Fifth one. But here methyl group is very important to remember for chlorophyll A, that is at the third carbon position. Third carbon position, and seventh carbon position you will be having 20 carbon in the form of a chain, giving the appearance of a tail for the chlorophyll pigment molecule, and it is called phytol side chain or simply phytol tail. So this is the structure of chlorophyll that is not clearly mentioned in your book, but for your easy understanding I made it clear. So chlorophyll will be having a distinct proper structure like this, in this manner. The pigment molecule will be having the center magnesium, and you can see the pyrrole chain also, phytol chain, and the rings, pyrrole rings are there. 5 rings are there. The first 4 are pyrrole ring, and the fifth one is called cyclopentanon. So the first 4 pyrrole are called as pyrrole rings only. The fifth one in the head one is called cyclopentanon ring. So the head is porphyrin head, which is tetrapyloric head, and the tail is having 20 carbon present in the 7th carbon of 4th pyrrole ring. Okay. Next. Excuse me. Next, what is the site of photosynthesis? This is the actual topic. What is the site of photosynthesis? Yes, photosynthesis takes place in the chloroplast. This is the structure of chloroplast. You are familiar with the outer membrane and the inner membrane. Always remember, simply it is not written outer membrane, inner membrane. There is some role how the reactions are going to take place, how whatever is formed in the chloroplast is going to come out, will be coming out through this membrane only. So you must have an idea about this outer membrane and inner membrane also. Keep in mind when you are talking about dark reaction, light reaction, there excuse me, you are going to discuss about the substances that are going to form and transported in the plant cell. So outer membrane, inner membrane, and the colorless part are the stroma. The colorless part is a stroma. This light green color is given here. This is a colorless part or aqueous fluid called stroma, where dark reaction will take place. And you can also see the green color stacks, these grana, these green color, these are called the grana, and they are one above other piled up. So they are going to have a thylakoid membrane here. This is also called the third membrane, which is a center for light reaction. Grana is center for light reaction, and stroma is center for dark reaction. Okay, light reaction means light will be harvested and water will be split to release oxygen, protons, electrons from the water. And meanwhile, ATP and ADPH are going to get synthesized, formed here. So in the light reaction, that is grana, the chlorophyll pigment molecules are present. In this grana only, the chlorophyll pigment molecules are present. And one more important structure is also there. We have the stroma, this one, stroma lamellae. Stroma lamellae is the extension of this grana. So this stroma lamellae, you can see it is present in the stroma, liquid portion, aqueous portion. So it is called stroma lamellae. And next, starch granules also you can see here. Starch granules and lipid droplets are also there. So the glucose which is formed, the carbohydrate will be converted into starch and lipid droplets, and this will be transported to their destination with the help of phloem tissue, which conducts the food. Okay, and next, one more important thing which is not given mentioned here, but it is present here in the diagram. This is csDNA. csDNA means chloroplast DNA. That means DNA is not only present in the nucleus, it is also present in the chloroplast. So it is called csDNA. And some ribosome also you can see here, ribosomes. These are for protein synthesis. These ribosomes are also found here. These ribosomes are also found here in the chloroplast. So this is the actual site of photosynthesis, which you are going to learn in this chapter particularly. And first of all here, absorption spectrum of chlorophyll A and B. So light is also very important. Light is getting harvested. Light is getting harvested by the chlorophyll pigment. So you must know how this light is getting harvested. The visible light will be having 7 colors, VIBGR, and you know the spectrometer will be there. The spectrometer, it is going to spectrophotometer, it is going to show which light is going to fall on the which light is going to fall on the leaf, and in the chloroplast, how the pigments are arranged. Different types of pigments are present in the chloroplast in the grana, and which wavelength light from 400 to 700 nanometer or 300 to 700 nanometer, as in some books, this visible light, which wavelength of light is getting trapped by the pigments. The pigments are going to trap the sunlight energy. As you know that yesterday itself I said, photons are the small energy packets. Photons are nothing but the light is going to travel in small packets called photons, and in this photon only we are having the energy. What is that energy stored in this? It is quantum energy. The quantum energy. This quantum energy, the more it will be trapped, the more photosynthesis will be taken. The less it will be trapped, less photosynthesis will be taken. That means rate of photosynthesis depends upon the absorption of this quantum energy. And this absorption of quantum energy is taking place by our pigment molecules. So here you can see, as you all knew, chlorophyll A, chlorophyll B, and carotenoids will take here. Absorption spectrum. Absorption spectrum is nothing but the quality of light, the wavelength. The wavelength we are going to take on a spectrophotometer, and we'll plot a graph. The graph will be plotted in this manner. And when we plot the graph, we are able to see chlorophyll A, chlorophyll B, and the carotenoids here. We haven't taken xanthophyll, we have taken only chlorophyll A, chlorophyll B, and carotenoids. You can see the dark green color is indicating the chlorophyll A, the wavelength. Here at this point, more at the blue color point or blue color wavelength, chlorophyll A is high, and it is less, and again it is at red wavelength, it is more. So this is about dark green color chlorophyll A, and olive green color is there. Chlorophyll B is olive green in color, and you can see this chlorophyll B is plotted here, and it is more at chlorophyll at this blue color only, and again it is dropping, and finally again at red color it is more. That means the blue and red color points, we can see more absorption is there. The absorption spectrum you can see. This is called absorption spectrum, and carotenoids also you can see at carotenoids also at blue color only. It is more, and then again at the red color we will see very less. So here carotenoids at blue color only you can see. So here in this manner the absorption spectrum is drawn for this wavelength, particular 400 to 700 nanometer wavelength at blue color and red color, the chlorophyll uh, is going to absorb more sunlight energy, quantum energy. That means rate of photosynthesis will be more in this area only. It will be known. And for this already I gave about Engelmann, and he has done for that green alga, cladophora, Emerson effect he has given. That will be dealt further now. So remember about this absorption spectrum. Absorption spectrum is nothing but how these chlorophyll A, B carotenoids are absorbing the light. In which part of the wavelength they are going to absorb more quantum energy, more light. That means more photosynthesis will be possible in that area. This is the action spectrum. Action spectrum is nothing but in this wavelength. Finally, by taking all those three, chlorophyll A, chlorophyll B, and carotenoids, we came to one conclusion. The action, that means the photosynthesis, rate of photosynthesis will be more at blue color wavelength. It is more at blue color wavelength and more at red color wavelength. And in the middle, it is somewhat less. So here, this thing you have to remember, action spectrum is nothing but the rate of photosynthesis when it is plotted by using the spectrophotometer. We can find out at the blue color and red color only, rate of photosynthesis is high. So with this, we came to a conclusion that blue light, red light, there the pigment molecules will be absorbing more sunlight energy. Next, action spectrum of photosynthesis superimposed on absorption spectrum. So superimposed means already in the first graph you have seen chlorophyll A, chlorophyll B, and carotenoids you have seen. So here the chlorophyll, you can see this one is the chlorophyll B, and this one is the chlorophyll A, and this one is the carotenoid. When we plot this one, this is called the absorption spectrum. This one, absorption spectrum. So these are the absorption spectrum. And if we see the rate of photosynthesis, that means action spectrum. Action spectrum. The action spectrum is indicating clearly it is at this blue color only. The rate of photosynthesis is more. Again, it is declining, and then again it is going to the peak level in the red light. So remember, on the x-axis, we have taken the wavelength 400, 500, 600 nanometer, 700 nanometer. And here on the y-axis, we have taken the rate of photosynthesis or the absorption of the sunlight. So be clear with these two terminologies, absorption spectrum, action spectrum. Absorption spectrum is the actual harvesting of sunlight by chlorophyll pigments. Action spectrum is nothing but after harvesting, they are taking photosynthesis only, no? So this action spectrum indicates the rate of photosynthesis. So it is more at this blue and red wavelengths. So I think it is clear for you. And here, one more important thing, Emerson effect or red drop effect, as I said earlier. Emerson effect or red drop effect is nothing but by taking the prism only, the cladophora, green algae was subjected to this spectrum, and it is plotted like this. And what Emerson has noted, you can see quantum yield. For example, quantum yield, if you calculate, it is nothing but the amount of oxygen released. That means after photosynthesis only, no oxygen will be released. So we have to know about the rate of photosynthesis. That means amount of oxygen released by quantum of light absorbed. How much quantum of energy of light is absorbed? That is going to give us the quantum yield. Means, sorry. So this quantum is nothing but the energy, light energy, sunlight energy. So sunlight energy we are taking, how much yield we are giving, that can be calculated, that can be known by taking into consideration about amount of oxygen released by quantum of light absorbed. This is very important. And Emerson effect is nothing but the same thing you are saying here, action spectrum. That means action spectrum, that is rate of photosynthesis. And you can see this one, green color is the absorption spectrum. That means the wavelength where more light is harvested. Okay, the wavelength is this blue color, and here red color. You can see the two peaks here. One is the peak at the blue color. Another peak is at the red color. You can see the red color also here. VBR colors are drawn here on the x-axis 400, 500, 600, 700 nanometer. You can see the VBR colors are also given for your clear understanding. So here we can plot these two graphs. Then over this we will calculate the quantum yield. When we calculate the quantum yield, calculate the quantum yield, then we can draw this graph, the quantum yield plotted here on this x and y axis. You can see here, and suddenly it is falling. Suddenly it is falling after red light. It is not having any quantum yield. That means only in this particular point after this red wavelength, it is stopping. There is no quantum yield. It indicates the red drop effect. Okay, this is called the red drop effect. So one more important thing about Emerson, he has given that Emerson's enhancement effect. Enhance means to improvise, means what is actually happening. For that, if we'll have certain strategy, we can enhance something, we can make it better. So here in this, you can see photosynthesis at 700 nanometer. For example, the quantum, here example is given, photosynthesis at 700 nanometer. That means at the red light area, you can see quantum yield is 10. And the photosynthesis at 653 nanometer, the quantum yield is 53. Now, if we take separately, it will be 10 and 53. But if we combine both these simultaneously, we do, then the photosynthesis at 700 nanometer and 653 nanometer combined simultaneously, the quantum yield is 72. Okay, that is more. This is called enhancement. By simultaneously shifting this photosynthesis activity at these two, 700 nanometer and 653, we will get the quantum yield as 72. So this is an example of Emerson enhancement effect, and this is because of two separate photosynthesis. With this, we can come to a conclusion. This is possible because at particular 700 nanometer, we are having one photosystem. Photosystem means a reaction center where the harvesting of light is taking place. Another reaction center at 653 also, you can see another photosystem is also there. Here, that is PS2. Photosystem 1 and photosystem 2 will be there. And photosystem 1 is at 700 nanometer and photosystem 2 is at 653 to 680 nanometer. So there are two photosystem. Depending on the identification, the name is given 1 and 2. 700 nanometer, it was found first. Then afterwards, the photosystem 2 at 653 to 680, there it is found. So remember this photosystem 1, photosystem 2. This terminology will be used in the further next session. Next, chloroplast dimorphism. So chloroplast is not same for all plants. Some plants, it is having a Kranz anatomy. Kranz anatomy means, you can see here, it is two types. The common type, as you already know, it is having the grana green in color, having the thylakoid membrane arrangement, and the stroma, the colorless. Okay, this type of chloroplast is called grana chloroplast, granum. Another one is agrana. Agrana means not having the grana. That means only it will be having the stroma only. There is only stroma. There is no grana. And you can see here, this arrangement is seen in Kranz anatomy. Kranz anatomy, the mesophyll tissue. You can see the mesophyll cell. Okay, and you'll be having a specialized cell called bundle sheath cell. Bundle sheath cell. These bundle sheath cell are nothing but they are present around the vascular bundles. As you can see here, this is the vascular bundle, and these are the bundle sheath cells. These bundle sheath cells are nothing but the specialized cells where you will be having the chloroplast. Okay, and the mesophyll cell. M is for mesophyll cells. So all over the plant there will be mesophyll tissue, mesophyll cells here in the center, and particularly the vascular bundle will be located here, xylem and phloem tissue. Surrounding these vascular bundles only, the specialized bundle sheath cells are present. This arrangement is called Kranz anatomy, and you can see how the chloroplast here it will be having a difference in conducting the light reaction and dark reaction. The light reaction will be taking place in the mesophyll cells because of the presence of the granum thylakoid. Okay, and here the stroma will be present, and here there are no grana. No grana means no light reaction, only dark reaction will be taking place in the bundle sheath cells. Okay, and bundle sheath cells are present surrounding the vascular bundles. Okay, so this is the thing that is called chloroplast dimorphism. One is grana chloroplast, grana chloroplast. Grana chloroplast will be having both the grana and stroma taking place where light reaction and dark reaction will be taking place one by one. In the mesophyll cells, only one type of cells, mesophyll cells will be present. Whereas another agrana chloroplast means the crans anatomy. The crans anatomy where you will be having the mesophyll cells. Mesophyll cells comprising of the normal chloroplast, mesophyll cells. This ash color is nothing but the mesophyll cell having the normal grana and stroma. Normal grana and stroma comprising chloroplast. Whereas the bundle sheets are surrounding the vascular bundles, and these are having agrana. Agrana thylakoid means not having grana, only having stroma and stroma lamellae. So I hope you understood chloroplast dimorphism. So here asked in UPSC mains also, you can see the vascular bundle is in the center and the bundle sheet is nothing but these bundle sheet cells are nothing but they are having a dimorphic chloroplast. That means they are having the crans anatomy, showing the crans anatomy, crans KRA and the crans anatomy having agrana chloroplast. Agrana chloroplast means not having grana, having only stroma, only dark reaction will be taking place here. And this one, you can see this dark green color is a mesophyll cell having the normal chloroplast. I hope you understand this topic very well. Next, here one more picture is also given, crans anatomy and non-crans. You can see the rice. In the rice, C3 cycle will be taking place, and the crans anatomy, the chloroplast dimorphism can be seen in the crans anatomy. That will be termed as C4 cycle or C4 plants, or the maize plant is an example for C4 plants. In detail, you are going to learn about C3 cycle and C4 cycle. C4 cycle or the crans anatomy having the bundle sheet cells having chloroplast dimorphism. That means having the mesophyll cells and bundle sheet cells. Okay, in the mesophyll cells will be light reaction. In the bundle sheet cells will be dark reaction. This crans anatomy, for some reason, it is developed in maize plant, whereas the normal rice plant, you can see C3 cycle can be seen. All the other plants, they are having non-crans anatomy. That means they will be having a chloroplast comprising of both grana and stroma. They'll be having only mesophyll cells, only mesophyll tissue. They'll also have vascular bundles as usual, but they are not having any bundle sheet cells. Bundle sheet cells are present only and only in this crans anatomy. Okay. Next. Okay. So we are now in assignment. Very short answer questions. Name the process which take place in grana and stroma regions of chloroplast. As you know, grana for light reaction, stroma for dark reaction. Where does photolysis of water occur? Photolysis of water will occur in the grana only because sunlight will be harvested in the grana only. And what is its significance? The breaking of water is very important. Water is split into protons, electrons, and oxygen. So, as well as when these protons and electrons are released, electron acceptors like NADP is going to become NADPH, and the accumulation of more protons and movement will make ADP to ATP. That you are going to learn ahead. Draw a neat label diagram of chloroplast. This is a short answer question. So this is the simple assignment given today for you. I hope you understood the topic very well, and read the textbook and have a clarity. Okay, and some points which are not there in the textbook are properly clearly dealt in this session. I hope you understood it very well. If you want to have a proper understanding once again revision, then you have to definitely watch the video which is uploaded. and make a note properly and give the feedback to your teachers. Submit your assignments in time. And thank you, one and all. Thank you very much. The session is over now. नहीं हुआ और खत्म क्लोज नहीं करी